Hello, I'm Karen Sime. I am the new director of Rice Creek. I've been here since September 1st, although I'm not new to experience Rice Creek. I've presented here a few times, as well as been an audience member and had my students present. And this goes back to 2008 when I was a assistant professor. And in my first semester, I got a Rice Creek grant, which was really a nice way to start out and gave me some critical support for my first field season here, working with swallowtail butterflies at Rice Creek. So I can testify to the big impact small grants have. They really help make research possible for us at SUNY Oswego. So it's really kind of exciting to be here as director and help support that aspect of it. Rice Creek Associates is the community support group for the station that actually gives the grants. And for those of you who are new, so they're kind of a critical support branch. In addition to the grants, they support the field station in various ways. They sponsor social activities, outreach to the community, various social events and projects like our new accessible trail, big recent project. So as far as this event goes, for those of you who are students or interested in doing research or creative work centered around the field station, I hope you find these presentations interesting. And if you are interested in doing research or an activity here that some grant money, you should talk to us, the presenters, about how we went about getting the funds, as well as to the Rice Creek Associates and the other staff. So, I'm going to turn it over to Scott Furlong, the provost of SUNY Oswego. Thanks for
Thanks, Karen. Good afternoon, all. I'm Scott Furlong. I'm the provost and vice president for academic affairs. Welcome. Welcome to Rice Creek. Really the not so hidden gem for SUNY Oswego, but given that it's a mile from main campus, not everyone always knows that we're here. I want to give a welcome to all of you to the SUNY Oswego campus. Red carpet.
welcome all of
I welcome you all. Anyway, I want to welcome you all to SUNY Oswego and this event. I try to get up here every year for this event. I think I missed one or two since I've been here as provost, and I really enjoy the opportunity to hear students, to hear faculty, their even community peoples present on what they're doing. I do thank the Rice Creek Associates for funding this activity and the support they give to the field station. It really is extremely important to what we're trying to do in partnership with the university. So with that, I turn it over. I'm going to turn over to Dean Kristin Croyle. So she can give her a welcome. Thank you. Enjoy it. And have a good day.
You all know what a delight today is, and it's wonderful to be together to see all of you. So for those of you who come, the Rice Creek is really close to my heart. And for those of you who don't, I'm Kristin Croyle. I'm the Dean for Liberal Arts, Sciences, and Engineering.
And Rice Creek is close to you.
And Rice Creek is close to my heart. A quote is attributed to Aristotle, who knows that there is something, all things of nature, there is something of the marvelous. And one of the things that I really appreciate about Rice Creek and the Rice Creek Associates is that you bring that forward to all of it. Where do we find that? In the spark of magic. You find the spark of magic in the way that complex ecosystems work together. You find the way the spark of magic in the collaboration between all members of the feel that spark of magic when you bring together students and faculty and people from sciences and arts. There is that. So I think today really brings that to mind.
I'm Laurel Artz, I'm the president of the Rice Creek Association. We really are a great team that works together and great leadership. And so thank you for being here. And thank you for this meeting. So people for being with us today. Based on the name, I mean, lots of email about this, so nice to this is fine. So I'll let you know a couple facts and figures. They're a little bit. I believe we now stand at 122 projects, totaling over $169,000. And that is since again 1996, we're going on. So pretty amazing. It's a great day to get together and see what's been going on. So we're here to support this issue. Always here, always. It really is.
I'm Katie Bishop. I'm first talk. I'm an assistant professor in the biological sciences department. So thank you so much for giving me the opportunity to share with you some of the work that two of my classes have been doing for the past 2 semesters. And so to tell you about that work, I'm first going to talk for a minute about our humble friendly earthworm. It may surprise you to learn, as it totally surprised me to learn, that the earthworms that we encounter when we go out here in central New York are not actually native to New York. New York does not have any native earthworms. The earthworms we encounter are a group of earthworms called lumberkid earthworms that are native to Europe. They came over with the European colonists when they brought over their crops and rootstock and soil material, and they established themselves in our local ecosystem. So for a couple hundred years, these have been an established part of our local ecosystem. So we would call this the first wave of earthworm invasion. But if you are paying close attention to the worms you see lately in your compost pile in your gardens, you might have noticed a newcomer to our worm assemblage here in central New York. These worms are actually native to Asia and Southeast Asia. They are more recent transplants. They came over with more modernized strains, same materials, rootstock, plants, etc. And so these have also invaded and are currently invading North America as we speak. So waves of invasion are spreading from where they're introduced to more remote regions. And so these have about a 50-year history here in central New York. They were first documented about 50 years ago, and they are marching across the state as we speak. So this is the second wave, ongoing wave of earthworm invasion. And so these worms up here are these invasive guys. They're called pheretimoid worms. It has to do with the genus that most of them belong to, as opposed to our more familiar lumberkid worms down here. There's a couple easy ways to tell these guys apart. The first is the patellum. So lumberkid worms, it's like this raised kind of saddle-like structure, but in pheretimoid worms, it's very flush to the worm. It's a very pale color. The easiest way to tell them apart is to handle them. So if you pick up a worm, the lumberkid worms will kind of wriggle, and you know how they behave. You've all baited a hook, right? These guys will like convulse in a very snake-like fashion and like propel themselves across your hand and across the soil. And that's where they get their common name of jumping or snake worm. That's kind of how they move when you handle them or they feel friendly. But the most striking difference is actually in how they interact with the ecosystem. So lumberkid worms, our older European worms, actually spend a lot of their time below the surface of the soil. They're moving a lot between the litter layer and below. These pheretimoid worms are mostly surface dwellers. So they're living mainly at the surface of the soil, and they're consuming mainly the plant litter at that surface. So the consequence of that is that we go from like our normal kind of healthy level of leaf litter in areas where these worms are really densely colonized. They clear that leaf litter out really, really quickly, and it doesn't accumulate. Furthermore, they're dropping their castings, which is a fancy word for worm poop that I recently learned, all over the surface of the dirt.
And as they're drying out, these very granular particles kept passing. They're giving the dirt this really like coffee grounds look and feel. And so you might notice, I've been noticing it everywhere I go now. And so if you like notice a patch that looks like this and you feel it, it'll feel like loose coffee grounds in your hands.
Every
And
year they
so
do
I'm
it.
going to pause on the worms. And you might be wondering, we're a microbiologist, you study yeast. Clearly, you just learned about worms. Why are you talking about worms? And
Number
so I'm talking about worms because the best way to learn science is to do science. Something I probably don't need to convince anybody of. This type of inquiry-driven learning has better learning outcomes for students, and they're also better and more engaged in the topics that they're learning. So inquiry-driven learning is when we teach scientific concepts by applying the scientific method as we go. So we're applying the scientific method to a real-world scientific problem to teach some approach or analysis or technique that we would use in science.
Welcome.
And I think we do a great job of this here as we go in the biological sciences department, but we all come up across very real-world constraints. There's only so much time in the semester. You have to have a project that can fit nicely in the time that you have with your students. Project setting data. And so the way I do this in my class is I leverage an approach called structured inquiry. And so I give the students the answer. I give them the method, the approach that they're going to use. That's what I'm training them in. That's what I would like them to learn. And then I try to empower them to interpret our findings and to contextualize our findings into the broader literature and to come back to the original hypotheses that we're testing and try to determine whether we supported or rejected those hypotheses. And so this works best when I have very straightforward experimental designs that can yield data very quickly and that I have really engaging questions that our students really get interested in and want to dive into. And with our student population here at Oswego, this often involves questions that gets them outside and gets them dirty and gets them touching animals. So
I'm not going to eat it all.
Right? So at this very event last year, I heard from Dr. Mohammed that there was a recent report of jumping worms here at Rice Creek, and I dug into the literature a little bit. And what I learned is that there is little to nothing that's known about the impact of these invasive worms on the soil microbes that live in the soil that they're colonizing. And so I thought that this would make a really great inquiry-driven question for my teaching labs, both microbiology and technology. And so RCA, through funding this, has enabled us to do this project. So I'm going to share with you a little bit about this project today. First, I'll share with you the learning objectives I have for my students. The first of that is that they understand the foundational role that soil microbes play in a terrestrial ecosystem. The second is that they understand how modern microbiologists collect microbiome data, analyze it, and determine microbial composition. And the third is that they understand the key limitations of those approaches. And so this empowers them to have a more critical lens when they're reading this literature and seeing these types of findings in popular media. So I'm going to treat these like mini learning objectives of the talk today. So hopefully after my talk, you'll be able to speak a little bit to these too. And so I'll start with understanding how soil microbes function to form the foundation of terrestrial ecosystems. And so if we go out and hike on a trail here at Rice Creek, we will observe lots of biodiversity, plant diversity, animal diversity. But most of you probably know that the vast majority of the biodiversity out here is beneath your feet in the soil, and it's hidden and you don't see it, right? It's the microbial diversity. And not only is this the vast majority of the biodiversity, but it's also critical functional biodiversity for the ecosystem. So this is forming the sort of the foundational part of the terrestrial ecosystem. And so to illustrate this, I'm going to give you one example, but it's a really important example. It's the example of nitrogen. And so macronutrient that we all need, plants, animals, we all got to have it. Most of the nitrogen on Earth is locked up as atmospheric nitrogen. Plants and animals can't use atmospheric nitrogen. It's just nitrogen. You can't assimilate it. So the only way that nitrogen gets into the food web is through being fixed by bacteria and archaea. They're the only organisms on Earth that do this, and all other organisms depend on them doing it to get their nitrogen. They're also the only organisms on Earth that convert nitrogen to other really important compounds for the nitrogen cycle, and the only organisms on Earth that return it as atmospheric nitrogen so that it can be fixed again. So we call this the nitrogen cycle. Really only this reduced nitrogen here and to a little extent nitrate here are readily assimilated by plants, and then that's assimilated by the animals that eat those plants. This is the form of nitrogen that plants and animals really, really depend on. So plants in particular are depending on these organisms to fix the nitrogen in the soil. And this relationship is so important that plants go out of their way to house and recruit these types of bacteria to their roots. And so they'll build little houses called nodules in their roots, and they pack them full of this nitrogen fixing bacteria. Some plants go even farther and they form partnerships with fungi in the soil, and the fungi go out and scavenge for nitrogen that's been fixed by bacteria, and they bring it back to the plant root. And those are called mycorrhizae. And so the fungi will bring back both reduced and oxidized nitrogen to the plant. And this is so important to the plant that plants spend a lot of energy recruiting these partners by sending out these messenger signals into the soil, these chemicals that are going to attract these microbes to the root.
That's
And
what I
of course,
had.
this whole thing, like any biochemical process, is largely impacted and mediated by abiotic factors. In the case of nitrogen, it's oxygen.
2 meatballs.
So, just with the example of nitrogen, I hope I'm communicating to you that there's a reciprocal feedback between the soil microbiome and all of the other organisms in the ecosystem. And so the soil microbiome is impacting the plants and their health and their diversity. Plants, in turn, are shaping the soil microbiome through the chemical signals and attractants that they secrete. But we know we're missing a lot in this picture here. We're missing the interactions of soil invertebrates, where they're both interacting with the microbiome and the plant systems. We're missing the impact of animals that are interacting with all of these levels. We're also missing the impact of all of those abiotic factors that are mediating what is chemically possible in these systems. And so if we step back and we look at this really complex reciprocal web and we remember our jumping worms, we remember the fact that the jumping worms colonize habitat that looks like this and they turn it into something that looks like this. And so they're modifying the nutrient input by eating all of that leaf litter and plant material. And they're also modifying sort of the physical properties of the soil itself. And so hopefully now it's reasonable to you why we might hypothesize that just the presence of this new worm might impact the composition of the microbiome. And if that's in fact true, we can speculate that that might have broader effects across this whole system. And so how did we begin to answer this question here at Rice Creek? The first thing we did is we picked focal sites to focus on, both that had worms and that didn't have worms. And so first we wanted a site that's been colonized by the jumping worms. And so big shout out to Dr. Kristin who marched all over Rice Creek with me one cold winter day and helped me pick these sites. And so our jumping worm colonized site is actually right here near the field station. It's right behind the equipment storage shed, and it's a stand of mixed ash and maple trees. And so I wanted to sample at the base of trees so that I had like a good landmark to return to the same place every time. And so we've got 3 marked trees, so it's sampling about 3 feet from the base of. And then we wanted a site that was not colonized by these jumping worms. So we had to trudge very far from the field station all the way out on the red trail past the stone wall to this wood lot out here, where there's another stand of mixed ash and maple. And we chose 3 trees to sample at the base of. So these were my control sites and these were my farm sites. Next thing we did was go about designing the experiment in terms of sampling. And so I wanted to both follow the life cycle of the worms, but also the academic calendar. So it could fit into my microbiology and my fungal biology class. And so we decided that we would sample the soil late April to early May. So this corresponds to when the juvenile jumping worms are just hatching and they're just kind of showing up in the soil. It also corresponds to when my microbiology class are doing their independent projects. And then we sampled again in September, which corresponds to, these worms that sort of have the full summer now to be in the soil, do their things. We're going to sample again at the end of that season. The adults are dying off. They've laid eggs for the winter. And this also corresponds to when my fungal biology class is doing their end of semester projects. And so we fit neatly into both of those calendars. And so in April, this past April of 2025, my microbiology class collected and processed samples. We collected soil and processed them for both fungal and bacterial diversity, but they only analyzed bacteria. And then in September, this past September, my fungal biology class collected and processed 6 samples yet again at those same sites. And they collected both bacterial and fungal. They are analyzing the fungal data as we speak. So I'm not going to scoop them too much today, and I'll focus instead on what my spring microbiology course found. So this is how we sample the dirt, pretty low-tech soil sampler. We get in about 1 centimeter deep. So we really want the top layer of dirt since that's where the worms are, and that's what they're sort of affecting here. Here is Kristin and Skylar. They are bagging up some dirt for sampling. So this is one of our control sites. You see we have to kind of rake and clear the leaf litter away before we can sample. Here's one of the worm infested sites, which is usually not covered with litter at all. And you can kind of see that particulate coffee grounds that I was talking about. It's really obvious when you get kind of into the soil. And so I was really happy that at our control sites, we found plenty of lumberkid worms. And at our colonized sites, we found plenty of jumping worms. So, you know, Dr. Haynes promised me they'd be there. They weren't back there. So that was good. We found the right worms. And so the next thing we need to do is bring the dirt into the lab and determine the microbes there. And so how we do this in modern microbiology is through sequencing. So my classes would bring the dirt back to the lab, extract total DNA from that dirt. At this point, we have two kind of options. We can go ahead and sequence all of that DNA. That's a really resource intensive process. What is a more common approach in microbiology is to sequence just parts of that DNA in a method called DNA barcoding. And so the best way to explain this is to compare it to, like, you're taking a trip to the grocery store, you're putting a bunch of different groceries in your cart, cereal, beets, pretzels, and those are all very different, right? But one thing that they all have is a barcode. So they've all got a barcode, and the barcode is different between all of those items. And when you go to checkout, the only thing the checkout person needs to know is the barcode. They need no other information to know what's in your cart. So we do the same thing in biology. We pick a gene that every organism that we're focusing on has, and that's a little bit different between each of them. And all we need is that one gene, and that tells us what organism we have. So we call that DNA barcoding. And so for whatever group of organisms we're looking at, we choose an advantageous gene that has those properties. They all have it. It's different between them. And we treat that like a barcode. And so in my classes, all we did was we amplified, isolated, and then sequenced this one single gene. We had to send it off because it's a next generation based sequencing technology. And through that, we could identify exactly what microbes were in our samples. And so for bacteria and archaea in microbiology, we use a 16S rRNA gene. In that spring 2025 sample, we identified 9,140 unique microbial taxa. In fungi, we use the IDS2 RNA gene. And in our fall 2025 data, our spring 2025 data, we identified 4,920 unique fungi. So these are the exact same soil samples, the exact same DNA prep. I'm just sequencing the bacteria in one of them and the fungi in the other.
I don't think it's
So
killer.
what this actually leaves us with, and what my students get back, is this very intimidating table where they've got thousands and thousands and thousands of rows, each corresponding to a unique microbial taxa. And then they've got a bunch of columns that tell them how many sequencing reads were mapped to that taxa. So this is a very difficult table to interpret simply by looking at it. So the first thing that we do is we give this to a computational statistical program to ask it to do what's called dimension reduction. And that's telling a computer basically look at this table and only kind of focus on the parameters that matter in terms of variation and reduce that down into two axes for me.
Okay.
And so here's what that data looks like. And so this is what's called a PCA plot. The axes themselves are kind of meaningless, and you don't need to think about the absolute value of them. What you want to look at is how close the dots are to one another. And so if the dots are close to one another, those microbiomes are really similar. And if the dots are really far away, those microbiomes are very different. And so here's the data for the bacteria in the spring. So in the spring, we got a pretty clear pattern where the worm colonized microbiomes were different than the control microbiomes. So they clustered together and they were more similar to each other than they were to the control. We got the exact same pattern with the fungi, even more strongly, where the fungal microbiomes for this soil clustered really closely, and the control were more dissimilar and also had more variation between them. Okay, so the microbiomes are different. Awesome. Next question is, how are they different? What organisms are different between these two microbiomes? So here I'm going to focus just on the bacterial data. And so we can pop this in to a couple different methods that are going to look at differences in composition, where they're going to look for taxa that are significantly different between the two groups. And when I do that, I get this set of data that tells me that there's, a handful of taxa here that are significantly different between the jumping worm colonized dirt and the control soil. And so here the purple bars represent taxa that are higher in abundance in the worm colonized soil. Red bars are taxa that are higher in abundance in control soil. I'm going to focus on those that are higher in abundance in the control soil for a second. So these are bacteria that we find in our control soil that we don't really find in our jumping worm soil.
Oh my God.
We found two bacteria that are obligate symbionts of nematodes. This is a totally different kind of worm, but it's It's not related to earthworms, it's a much smaller type of worm. Still lives in the soil, still has really important interactions with plants. And so what these data are actually telling us is that it might be that these nematode worms are being displaced in the jumping worm soil because we lose their symbionts in this soil and we still have them in this soil. So that was the first thing that stuck out to me is maybe there's a three-way interaction going on here. The next thing that really stuck out to me is we had a whole bunch of groups here that belong to what are called the rhizobiales. These are nitrogen fixers. So that whole nitrogen cycle I showed you where they're grabbing the atmospheric nitrogen and they're giving it to the plants, these are present in high abundance in our control soil and they are depleted in our jumping worm soil. So something's going on with the nitrogen cycle here where we're losing these nitrogen fixing bacteria. When I look at the groups that are enriched here, there's not a whole lot of biological signal. And what's really unfortunate and what is a key limitation of these types of studies is oftentimes when we have a barcode, we can assign it to a microbe, but we know nothing about that microbe. We've never cultured it. We don't know anything about it. We don't know how it behaves. We don't know any ecological information about it. We just literally know it exists. We call it biological dark matter. Most of these are biological dark matter. So I know that they're different. I just can't really say anything else about them. So the final thing we did in micro is we assessed just microbial diversity. So how many different microbes are in these communities, and it's a difference between these two ecosystems. So that's the final data I'll show you. The first measure here is species richness on the right. That's just the total number of microbes. And you'll see that there's like a little bit of a trend where there's more microbial species in the worm colonized earth. Probably not. However, when we bop over to diversity, that's not just how many microbes are here, but how evenly spread are they in abundance, right? It's just one taxa dominating, in which case that's pretty low diversity. Or there are lots of different species in high numbers, that's higher diversity. There's a pretty clear pattern here that the presence of jumping worms increases the diversity of the microbiome. And you might think, oh, that's good, right? Biodiversity is good. Yeah, usually for trees and birds, it's good, right? In soil microbiomes, it's actually not. So enriched microbial biodiversity is a signature of disturbed landscapes. And so when you disturb a habitat type, you increase the microbial diversity. And really pristine habitats tend to have very low microbial diversity. And so this is a signal that this is a disturbed system, an active soil. So I just want to wrap up the talk by circling back to the learning objectives. Hopefully I've been able to demonstrate how I meet these with my students using this project. And then I just want to thank RCA for funding the project. Special thanks to Dr. Haynes for helping the site. And then these are the students that out the project. In micro, we have Zoe, Caitlin, Keith, Noah, Aurora, Bryce, Sierra, Jenna, and Jackie. And then in fungal biology, Skylar and Christy are leading the project. And I guess yeah, I think when we dig deeper, because we're still not done analyzing this data until my spring class gets it in microbiology. I think when we dig deeper, if the nematode signal is still there, I think that is something I'd be interested in. I don't know how to look at nematodes. We probably don't capture a lot with our actual soil sampling, so I couldn't use yeah, not really. So I'd probably have to learn more about nematodes or collaborate with somebody. Yes, about nematodes. I'll want to see. Absolutely. I think these people yeah, we could go and look at their, so we don't know anything about them, but we do have their genomes. And so what we could ask is, what genes do they have? Do they have genes for glycolysis, for photosynthesis? Do they have genes for nitrogen fixation? Do they have genes for producing secondary metabolites? What are those genes? And we can speculate about their function in the absence of actually measuring their function. So yes, that's a great, and that is something we could and should hopefully do in the future. Great point. Yeah.
Okay, so we're talking about how this second group of worms and how it's affecting the environment. And the original worms that came from Europe actually did the same thing. They came.
Presumably. Yeah. No one was here to sequence the microbiomes at the time, but yeah. So that's, like this happened already and it all evened out, right? Or we're all just in an altered system and we don't even know that it's altered.
So
right?
there might be hope here.
Yeah. Totally.
Next we have my colleague, Sime and Machara Malone to talk about their study of butterfly diversity in Rice Creek Field Station. I think out of the three, it's kind of obvious that I'm Mike Coley. Not Karen or Sarah. This study has spanned 30 years. It started in 1996, and it was actually a result of a student who was in an entomology class here for exploring nature. And what happened was that I happened to teach that course that he was in. But what happened was he aged out of that course, good for about 2 years. After he left it, he wanted to continue looking at butterflies and collecting butterflies. So his dad was a professor here at the college, and they got an idea of canvassing all the butterflies that are found at the Rice Creek Field Station. And they started that in 1996. So in 1999, here we go. This is what I wanted. I think I got it.
I'm gonna see if this works. Yeah, take this now. Good.
It's even frozen.
Okay, I can imagine touch on that. So here is Dr. Weber and his son Meg, and like I said, they started in 1996. Nick went on to graduate, went to college, and I joined the group in 1999. In fact, when they started to do this, Pete had given me a call and asked me if this study had any merit. And I said, sure it did. So they went ahead and applied for the RCA grant. And after I got off the phone with them, I said to myself, people tell me, why did you think of that? Because I've been doing this all, I've been collecting since 1964. So, but anyway, I joined them in 1999. And why do we do this? Okay, determine the species present, of course. Establish a phenology. When do certain species first appear? What's the last date that you normally see them? And then there are indicators of environmental health and biodiversity. Of course, the more diversity in butterflies you have, the assumption is the more healthy that particular environment is. There's a fourth one that's not up here. It's fun. It's just fun to do. Okay, so we did this study using what is called a Pollard protocol, and Karen will talk a little bit more about that later. But basically, it's a standardized way of going down some determined transects and just sampling what you see in Q5. So we mapped down areas on blue, green, and red trails. We had approximately 17 transects or so that we did. And it's about a 3.5 mile total distance walk, which is nothing for me. I normally walk 3 or 4 miles a day, even when I'm not at the station. And over here you see, we just basically mapped out our transects and made the numbers for the blue trail, green trail, red trail, and then some of the fields. So we did this survey once or twice a week, starting in May, ending in October, and basically from 10 to 2, observed the number of hours, the species, number of individuals, any kind of photos, to try to get photo documentation of every species that's here at the field station. And on the close to 70 species that we've documented, I don't have photo documentation for just 2. So we're doing pretty good. Species number of individuals, okay, behaviors reported, and the fact that we're using this Pollard protocol makes it standardized so that we can compare things that we're doing in this part of the country, other states, United States, and even across the world. It's a very standardized protocol. Been very depressing over the years because we found a decline. We found close to 70 species. We don't see all 70 species every year. In the early days of this study, we were averaging anywhere from about 40 to 48 species a year. Most recently, it's averaged out to about 30 or 35 or somewhere in between there. I did have one year, a couple of years ago, where I got up to 40 again. But this year was a very outstanding 26 species. The number of, not only have the number of species gone down, but the number of individuals within those species. So as an example, if we're looking at monarchs, so we see monarchs every year, but we don't necessarily see the numbers like we used to see in those. Even of those that we do see, the numbers are much lower. Some are no longer seen. We've got a few diagrams coming up in a second. Hair streaks and euphalics. Hair streaks are small butterflies that are like this. They're kind of little tail on the end that looks like a piece of hair, which is why we call hair streaks. There were 4 species that were found here, the 90s and early 2000s. I see none of them. I think one of them might still be around. Saw one maybe a couple of years ago. It landed on the windshield of my car. I think with that particular one, they live more up in the trees. So unless you go tree climbing and try to go through these things, which is very practical, you don't normally see them. So here are some things that we used to see that we don't anymore. One is a European skipper, which isn't invasive. We used to see them, quite early on in the study. I think maybe around 2004 or so was when we stopped seeing. Harvester butterflies. The caterpillars of the harvester butterfly are the only caterpillars that are true carnivores. Feed on woolly aphids. Any other caterpillar, any other butterfly usually feeds on some kind of. These guys are true carnivores. In Baltimore checker spot, which happens to be the state butterfly for Maryland. We used to see these particularly in the middle field during the month of July. I haven't seen any in such a month. Then this is an example of a hair streak. The banded hair streak used to be the most common hair streak here. I went looking because we have a lot of ash trees that met their demise to emerald ash borer. And I went to look at all the butterflies to see what their food plants are. There's only a couple of butterflies that actually fed on ash trees. And I don't think they were really impacting because they feed on other plants as well. So that shouldn't be too much of a difference. But I was particularly interested in the banded hair streak because I used to find them around ash trees. The caterpillars of the field. So okay. This is where you come. Yes.
So this is where I come in to the project, the color-based butterfly network. So starting in 2012, the so-called North American Butterfly Monitoring Network was founded to promote collaboration among people like who've been doing these surveys mostly around North America, although there have been some going on in Europe as well. But this one is particularly aimed at the North American surveys to get them to systematize their monitoring methods. They haven't already done so, and then start working together to collate their data. Right now, as of this year, the Butterfly Monitoring Network includes 36 general survey sites, of which Rice Creek is one. general survey, meaning that we survey all the butterfly biodiversity in a given area. And there's also 13 monarch specific sites that just monitor monarch butterflies that have joined this effort. And in the last few years, what this group has done is they have, supported by the National Science Foundation grants, they've developed a database, which all of these survey sites, the personnel can enter their numbers into. And a few rules here: the data collection that goes in, the data that goes into the database must have been collected following this Pollard survey protocol, which for Rice Creek worked out great because they've been doing it all along. Okay. Some professors at University of Maryland and Georgetown University have been organizing this. And the cool thing about this is that it's giving us a really strong data for analyzing butterfly population trends across regions of the US and across North America. And it also is linked to some European groups that have started doing some of the same things. There's been a lot of talk about insect decline in recent years and butterflies in particular. And these sort of efforts are where we can get really good data to try to really see what's going on. Okay. So the Rice Creek grant that we got last year that feeds into this, our idea here was several-fold. For one thing, we wanted to bring students into the project. Mike had been doing this mainly on his own in recent years and thought it would be a great idea to bring students into it. And for that, he wanted to collaborate with a professor who would turned out to be me, so I'm the local entomologist, and I have some experience with butterflies. Okay. It would be a great way, we thought, to teach students field survey techniques as well as working with the database. And one of our motivations for that was that it has a lot of data. So 30 years worth of data, and every year there's 20 to 40 survey days. The average is around 30 each year. And each of those days has, 4 hours worth of data. Sometimes there's hundreds of butterfly sightings on those days. And it takes a long time to enter. So we had this challenge that Mike wanted to get all of that data into the Pollard base so that we could contribute to their long-term analyses. But that itself was a very difficult task. So he wrote me into doing some data entry. And also we wanted to bring some students on board with that. So that is why we wrote the RCA grant, was actually just to pay a student to do some of this work with us. So kind of just showing you what these data look like. This is one of our field data sheets, and we use blue or green paper so that the sun doesn't blind us when we're writing on it. At the top, you can't really read it, but we record things like date, time, weather, who's doing the data collecting, what time they started. And this mess here, this is my handwriting, which is not great when I'm walking around, are the names of the butterflies, number of individuals, which trail it was on, so blue trail, green trail, and so on, and which section of it, and then some notes about habitat, whether a picture was taken, and so on. And then Mike enters it into Excel, and then we enter it from that into the database. I'll hand off right here, because that is where she came in for the database entry.
Yeah, so I started working with them in like March or so, and Mike sent me very detailed instructions on how to enter into the database. It was super straightforward, but I gained a lot of experience from doing the database. I found that it's helped me in my other courses because I've been doing a lot of other data input because I'm a senior. So this is what the website looks like. Every time I go to enter, you go to the trail name, and then it shows like the portion of the trail. It shows that this is the blue trail. And then for every entry, for every trail, there's put in all the information that was shown on the Excel sheet. And at the end, it shows a summary of all the different types and the numbers we saw. So it's a really good way to just see everything together. And then in the field, when I had a chance to go out with them, it was really awesome to experience field research. And Mike is an expert and Karen is an expert too. A partial expert. Getting there. I think she'll get there one day. But yeah, it's been really great to work with them. They're both lovely, and I've learned a lot both in the field and doing the database. I've learned to be a lot more efficient in data collection.
So we plan to keep this up. I mean, we're not going to stop in 30 years on the web. But we will continue doing the surveys, and as are suggested, like this training needs to do this. Although my work so far in graduate school, I worked a lot in small scale butterflies, but I still depends on a lot of this still working on that. Yeah, getting there. What I do is I take pictures and I send them to my say, what is this? That's usually how it works when I'm doing it about. So as far as the database entry goes, it's extremely tedious work. Would you agree with that?
Yeah.
Yeah, it can be a little bit mind-boggling. So in this last year, we've entered about 5 years worth, okay? Mostly done by Machara. And so it's going to take a few years to get that 30-year data set into the Pollard base, okay? So for that reason, we'll continue to work on it. Mike and I would like to continue to involve students in the project. I think it's a really great way to learn about some of these methodologies. By taking students out in the field and then working them through all the data entry into Excel and then into the database, it gives students a really kind of full experience. They start with the natural history and ecology by going out with us for hours in the field and looking at butterflies, and also how the data is acquired and managed. You know, we know something about the decline of insects worldwide. We know about the impacts potentially of the climate change and insecticides upon insect populations, because there have been some of these long-term, large-scale data projects in various parts of the world. And Rice Creek and other field stations are actually a really important component of that, okay? Because we provide a relatively stable environment in which data can be collected over 30 years without someone coming in and like logging everything or something like that. Yes, there have been some natural disturbances to Rice Creek. Most recently, I think, is the death of all our ash trees. But overall, it's more stable than a random environment. So field stations play a large role in providing data that informs conservation practices. The other thing that Mike and I are interested in is spin-off research projects. When we get students involved with this, they often, they often think about different little research projects that we might be able to do with this. And some of the things we think would be great small-scale student projects for a summer or a semester would be things like looking at the diversity on alternate routes. If no one ever does the Orange Trail, okay? Maybe that would be worth surveying. Or looking at different times of day. We have a set pattern, start with the green trail, then we do the blue trail, then we do the red trail. It's possible that some of our results will be different if we change that up, and I'm curious about kind of checking on some of our survey methods just to see that. Larvae, we don't really look for larvae very actively, like this monarch larva here. We take down notes if we see them, but we don't really survey the larvae, not actively looking for them. So that could be a nice student project as well. Other things like the behavior, looking at which nectar plants are used by the butterflies over the course of the season. And then also, as we get this data organized and into the database, there's a lot of data there that finds for a student project. So for example, Machara is actually doing this. She is for her wetland ecology, right? For wetland ecology, she's doing a little mini research project in which she's looking at one of the wetland species of butterflies. It's the leaf skipper, which we see mostly over on the boardwalk of Green Trail. And she's looking at its numbers over 30 years, abundance and phenology. And that was the main thing you were looking at, right? It's sort of like when their seasonal peak is over time, see if that's changing. But that's a work in progress. So I can't tell you yet. Maybe next year we'll talk about that one. And I think the project, besides being a great training mission for students, has a great overall value. It's part of this worldwide effort to understand butterfly, in particular, and insect populations in general over time, and to see how, for example, climate change and other environmental disturbances, habitat loss are affecting them. This is sort of the raw data that those kind of projects need. And it also raises the profile of rice creek. We're part of this nationwide and to some extent worldwide network of butterfly surveys. So we'd like to acknowledge RCA for providing funds for this project in the last year. The Rice Creek staff has helped a lot with keeping the trails open and keeping the habitats in good shape. So Kristin, Camille, Wendy, Bill, and Tom have all supported this project in various ways. And Jeff Pippin is in contact with a Pollard-based project who has basically set it up for us to enter and kind of keeps life in line with the various methods that we want to be used. So I'm going to get in there, and if you guys have any questions, I can answer them.
It must be, even.
Yeah, the other survey sites they're actually entering their data now into the same database as RCA, so it's all part of the same system.
There are also separate studies being done besides this butterfly monitoring network here. I have shared my data with people in Great Britain, Finland, and Germany. They're doing more of a larger worldwide study, and they've received all the data. Thank you. Machara, why did you do this?
Well, I wanted to get involved this summer. Just doing something zoology, and I do happen to love entomology. Go entomology. But and I saw there's an email Dr. Sime sent out, and I just replied to it, and I said I'm interested, and then I met with them.
Thank you for doing it. Mike, what was the last time you discovered a new butterfly species? Oh. Out of being at least a few years, there is a skippers look very, very similar. Okay. And there are three skippers whose females are known as the three witches, okay, because they all look so similar. But I've been looking at this data a little bit more, and there is a fourth one that I think I've been misidentified as one of the three. And I can't think of the common name. It's oryzivorus. And I'll be looking at tons of photos in the near future and to try to separate that guy out. It's the females that are the colors. They all look the same. They all look the same.
What's the rarest one you find? It really surprises you when you're sticking to see it.
There are so many that I'm tickled to see what I see. Compton tortoiseshell was one. Milbert's tortoiseshell. There's a brown intelligent, big eye brown, northern curly eye. there are so many of them because we're at a state now where we see so few of these. And even if we haven't seen them after, let's say, a span of 10 years or so, and it happens to show up again, and it makes your day. Sometimes I'm ready to say, I don't care if I could be out on the Great Trail and spot one of these things. And we have 2 more major trails with a number of transects in each one. And I'm saying, I'm so glad I saw this one. If I don't see anything else, this made my day. It's just, it's the thrill of the hunt going out there. You never know what you're going to see. A transect is just a certain trail section on the best spot, and we just monitored. But for instance, the Machara was talking about this briefly. On the Blue Trail, we have 4 transects. So we call it B1, B2, B3, and then we have a B4A and a B4B. And the reason why it's split is after B4A, thank you. We start with a set of field transects that goes zigzag across the blue trail. And when we end that, it comes up to the rest of the B4 trail. So we call it B4, B4B. Yes.
Pardon my novice question, but you butterfly people email each other internationally. You mentioned you were in contact with others.
I had been a member of the Lepidopterosis Society, which is a worldwide thing. They have a convention every year here in the United States. 1 year it's on the East Coast, 1 year it's on the West Coast. I make practically all of the East Coast. Once in a while, I get out to the West.
Thank you. You read my mind for the first question. And the second question is just do butterflies hitch rides really easily the way other species do? They can travel internationally. Oh, they travel internationally. How can they survive those long trips? Because they need constant, more than others, need constant.
Well, it depends what stage they're too. They could come across as an egg. Okay, it could be a large, it could be
so they can survive that.
Right. There has been documentation of butterflies like peanut lady flying onto a ship that's in road somewhere.
They get blown around.
Yeah, that one is known as the cosmopolitan. It's found all over the world. We had a really big explosion of them here in 2017. Right at the time we were experiencing a monarch migration. And they were all over the place. I think on one day I documented over 400 of them. And I think I skipped. I missed just about as many. I was counting them by 5s and 10s. Also counting the monarch migration, in addition to the other ones that we normally see. What an exhilarating day. I was exhausted. Well, it was just a marvelous thing. And to make things a little bit even more interesting, I forgot my field data. I didn't have the pages to write on, so I came in here to the field station. I borrowed some colored paper, and after 3 entries, I drew a line across because I don't write straight across. So I wanted to make sure I didn't get things stuck. So every 3, I would put a line across just to keep track of it. Yes.
Over the years, secondary stream, some species appearing earlier or later in the season.
Yes, and one of the problems with that too is we might see the butterflies, but we don't necessarily see the flowering plants. So milkweed would be a good example. Milkweed is mainly in July. I've seen some butterflies that are appearing June now, where milkweed is out. So they lose that as an image. So there are things are all disrupted. I happen to believe in global warming, so I'm sure that is a factor. One of the questions that I came up with was, if the butterflies are experiencing this, why aren't the pigs? And they probably are to some extent. We just need people to study it. Oh, yes. Yeah, I'm running another engagement later on today, and I have to set up.
Thanks. Before we let them go, I just want to make a comment that it's really hard to sustain a project for 30 years. And this is a tremendous accomplishment, and there have been many people including are involved, but so much of this is on Mike. And so thank you, Mike, on behalf of the field station. This is really elevated. And as we know, we're in this insect biodiversity crisis, so we need these data, these long-term data sets. And a very other side is that I stepped out, so I don't know if Mike mentioned this, but he has a wonderful book recounting the species of butterflies that's found here with stories about them. It's called Wandering Wings, and it's available at the River's End in town. So if you're intrigued, learn more about this survey, my experiences with butterflies.
We can't fly away.
Our next speaker is Professor Kelly Rowe, who is the Associate Dean for the School of Communication at Artz. And she is here to talk to us about her photography project, where she photographed insects and made a photographic essay of insects and environments.
I'm Kelly Rowe. Professor in the Department of Art and Science, and currently the Associate Dean for the Pollard in the Arts. And I'm an artist. So it's really fascinating to be here and hear the cool presentations that already happened. I'm intrigued, but I'm coming in with a completely different perspective. When I saw the Rice Creek grant application, as a professor, administrator, it's not always easy to find the time to create work. Artistic disciplines can be quite time consuming. So a few years ago, I went back to my camera and picked it up and started photographing things. I've always had my camera with me, and I drive my family crazy because when we do go on walks, they're way ahead of me because I stopped and I saw something move in the grass and had to photograph it. So it's been a really great outlet for me. The past couple of years, I've done a couple artistic projects. One was a book that was a collaborative book. I'm a bookmaker, photographer, printmaker. I like to combine all of those disciplines. So the project that I did, it was a call for, a walk in the wilderness was titled, and the only stipulation was a certain size of piece of paper with a horizon line for all of the artists to work at a certain date on that paper. And you had to go take a walk in the wilderness. And that's what I really enjoy doing. So that project led into photography challenge. There's a lot of challenges out there for photographers to do like a weekly theme. So I spent 2024 every week doing a photograph based on whatever this theme was. And at that time, I had my camera with me. By the time I woke up till the time I went to bed at night, my camera was next to me. I have a Nikon, which is kind of heavy, and then I'll have a few different lenses to go with that Nikon.
And I got really fascinated in that year with close-up photography. So I have a couple macro messages and events that I got through the grant here. And decided to do it last year when I was out here in the photographic specs. Maybe that's what I should do for this project. So there is a couple books circulating that has some of my favorite photos from my project. I learned a lot, and when I wrote my midsummer summary, one of the things that I was most surprised about was getting out of the office over here in Rice Creek and having to settle down. Right, you're in the office and every day in the associated team comes up with all sorts of different problems to solve. So coming over here and having to look for insects when you're looking for them, you can find them. So walking a trail and just stopping for a moment and looking, or just sitting down for a moment. I did end up with some kind of crash my second week out here. I got too close to some plant, but it did feel like a lot of things started to emerge. And that's when I felt myself calm down in the office time for, sometimes I was out there for an hour, sometimes it was 3 hours. And then, camera discoveries. Like, I found such a range of insects that I didn't even know what most of them were. Spend a lot of time trying to look up, and in that book, like, if you see something that's mislabeled, please circle it, right? This is not that. It's and it's good to hear the last presentation that, I'm close, but not spot on. This one too, that dashboard, the emerald dashboard, I thought, wow, what a beautiful insect. And then I got home and I discovered, oh my God, I should have killed that thing. And with my project, I proposed photographing 1,000 photos, not 1,000 insects. 1,000 photos. I think I got close to 2,000 photos over the summer. I also spent some time in the collection. Still wanted to see more in there. Some of the photos that I did take, I wasn't happy with because a photographer, the lighting has to be just right in order for you to be happy with what you've created. And natural lighting is good for me. Setting up lighting, I'm the best. So I do have a little work to do there, as well as identifying the creatures that I have found in mind. So, what's next? I don't know. It's really fascinating to hear kind of the scientific presentations and what we do with all of it, or what things I have found that sparks the interest on that side. And then I started thinking about our science students and how they think that, because I know a lot of them from fascination with nature and wildlife and insects. There's a lot of overlap there. I was in slides with thank you all.
Really beautiful work. The way you approach this perspective that you mentioned. Thinking about ways to use your observation more scientifically. It seems to me that the most straightforward way to start something for us, which is
I did see that, yeah.
within database where people can just submit observations. And That would be really
Okay.
nice thing about that is people can also help you identify. So you don't necessarily have to know what it is, you can make a guess, and people will check it. Like, that's the wrong part.
Right, right.
So you should definitely.
Okay.
Or like photos with other like animals, like birds or reptiles, and out that kind of thing. I find all of that really fascinating. And then even, you know, would love to go out with like, you know where they are.
It's hard to count and write down and take pictures at the same time, so.
I mean, I'm just wandering around like, oh, let me try this trail.
Thanks for your presentation and beautiful work. Was there any insect that just doesn't like the picture taken? Another point, did they fly away before you could get them?
Yes.
And then did you ever have to say, I'm so desperate, I'm gonna stay camera?
It
and
might,
be ready for them
it
to
might be
land
in your
that
ear
week
now.
and then-
Yeah, there are quite a
I can't hear you.
And, you know, I have that little green fly. I have probably 200 photos that are just blurry, nothing there, because they just are so fast. This one, this moth was really difficult for some reason, even though it was staying still on the bark. There's something about it that I just did not.
And I'm gonna detail, I know pixels aren't the entire metric you use, but roughly what pixels do you?
I have a close-up lens with my Nikon, so I want to say when I shoot raw, my file size is around the place. So a lot of these images are cropped with that close up. And some of them might look a little pixelated. There are a lot of discoveries of things that I didn't see when I was photographing, but then when I was going into the file and saw, like, wow, oh my God, there's so much more
Did you
here.
ever choose to use a phone camera or would they all be off the phone?
These are all on my Nikon.
I'm really impressed. It's quality.
It was, yeah, I printed those the other day in our office. So I just made sure that the paper is really important, the surface of the paper, and then the file size, that I have enough space for it to do that. Yes, I did.
You did that?
Did that.
Yes.
Oh, yes, yes. I love a paper.
So are you any thoughts about someone? Nothing experiments.
In my project too, I was doing photos, screening and options. It's just they're so beautiful to look at and do the process. I will tell you, 1994, when my girls was having grown up with school masters, I wanted to see our connections with partners and. Okay, that's a little nervous thing. Oh my gosh, it's hard to articulate. Take pictures. Thank you.
That they talked about the birds, you have, two biology students, working with Dr. Baldassarre, talking about, bobolinks, the fields around, Pollard area. So we have Shaylynn Schumacher and, Schumacher and we have, Monty mentioned, I said, right?
Yeah.
I'm Shaylynn Schumacher. I am a sophomore zoology student who studies, I have two research, I guess, like research I work on under Dr. Baldassarre. I also helped fund the northern saltwet owl banding that is pretty popular right now, along with completing being one of the banding assistants at the Rice Creek banding bird station over the summer, and hopefully I will get my hands more on more bird stuff because I'm kind of like a crazy bird lady.
Hi guys, I'm Julina Almonte, I'm a senior of biology. I just kind of told about my story each day in summer as well as research. And it was a really fun experience. I learned a lot, and I'm not necessarily a bird person, but I love reading about them. It was a funny experience.
They're definitely full of personality once you start getting there, getting out there. So, like as many of the previous presentations have covered, biodiversity, in addition to like, global climate change, has impacted what is locally around, and whether that be for birds, for reptiles, amphibians, butterflies, and all other species. I personally really wanted to focus on the conservation and the monitoring of bird populations. And since about 2019, it has been officially said that statistically speaking, avifauna has been increased by 29%, which has probably increased a lot since it's been a couple of years since that study has been conducted. And by monitoring the species, we're able to get like more information known about where, like, what habitats are preferred, or like if you're looking at like setting up nets like we do at the RCA banding station, what nets are like in a more ideal habitat for what species, and just understanding like productivity of this different like bird species. So we kind of did a lot of different bird stuff this summer. Notably, we were invited to Full Middle School around mid-June to promote bird banding and teaching, our younger generations about how we go about these processes. Because when you first figure out that you're putting birds in bags, sometimes it's a little jarring, but it can be really, really informative to all people. So we did go out to Full Middle School, teaching like what experiences you can have at Rice Creek, and talking with younger generations about what information we're taking, which we'll cover later in the presentation from these birds. But Rice Creek, our banding station specifically follows the monitoring avian productivity and survivorship, or otherwise notated as MAPS protocol, which has, it's a seasonal protocol that covers typically spring, summer, and fall, and it kind of depends on the season where we're setting up the maps, because otherwise the birds will figure out where we're putting the nets, and we kind of want to keep that passive netting kind of random, to really just like leave it to fate on what birds we're kind of getting. This year in the banding seasons, we had a total of 68 different species. We caught a total of 772 birds from spring to fall, which included 547 new birds that we banded. And we also do take information, the same information from recaps, which we get 225. Typically, when I didn't get a photo of the scribing sheet, but typically when we are doing like bird banding, you're taking information such as like vocal protuberance, the wings, like how big the wings are, Their fat, their weight, depending on what is happening, sometimes we might pull like an arcane feather to send out to other, like different stations or like different universities who are conducting, research on like maybe it's mites and what their relationship might be with the birds. And yeah, we kind of take a lot of information. We tend to get up over the summer. It was around for the banding station, like 4 a.m. And we do 30-minute net treks. And we typically run till about 12 to 1 in the morning. We have a total of 10 nets that go up. In the spring, I believe the nets were set up 1 through 10 in the like little
In the power.
in the power line. Yeah, couldn't even think of the word. But it was we did alternate the nets and move them further into like the hiking paths and stuff to ensure that birds weren't figuring out where we were trying to set up our nets and stuff. This is a very beautiful male indigo bunting that we were able to catch and get some information from. And that's fine for the camera. We did actually, shockingly, of course I missed this day. We caught a woodcock, which, if you're a bird person, is like a big deal. And this is kind of like what the setup is when we're taking any information. We have anywhere between 1 to 2 birds going, maybe sometimes 3. And we do tend to see a lot of like great catbirds, or cardinals. Yay. They're a little scary sometimes. And yeah.
So our research in terms of the model focus was on stability and add animals revisiting areas that they were referenced. And we were looking at the success of the models. So we should probably discuss what our model is. And to note, this was one of their nests, and this is kind of here, those are finding.
Yeah, unfortunately, it did get kicked out of the nest, so we didn't actually get to see what it was, but it could be a possible blue parasitism going on, which was really weird and kind of fun to watch their reactions to it.
Yeah, so what are bobolinks? So they are a type of songbird, and they are migratories. So they travel 12,000 miles to round trip. In the winter, they try to stay at places like Brazil, Paraguay, Europe, and then they would travel states like Virginia, first New Eastern Washington, and even places like Montana, Arizona. And this is where they breed during the summer. In addition to being songbirds, they are also grasslanders. They and as Shaylynn mentioned earlier, we have seen large decrease. Since 1970, there's been a steady. Well, in terms of grassland, population this summer, 7 million birds lost in 1970. So this relates a lot with their habitat. So as mentioned, they have habitats. These are areas that are not dominated by. It's so less than and this is one of the most important things like medicine, have been converted into farms and have been impacted by raising fire suppression. This can negatively impact bird population because now are no longer being able to survive. So with that being said, all of these things That's really grasses. But this tends to be hard to find, so they had learned to utilize things like the and this allows them to kind of have that. That's pretty cool.
Alrighty, so here are some photos that we took over the summer. Unfortunately, females are really hard to spot. They kind of like to sit lower in the grass, but when we're going about finding and identifying what our birds are, they are identified by 3 colored bands. And typically we're looking, the males are the are kind of the ones mostly making noise and singing. Baldassarre explains it as like kind of like R2D2 from Star Wars, which yeah, it's a lot of fun, and I honestly really miss like going out really early in the morning and listening for that. But the males, as seen in the upper right corner, are in the early seasons typically chasing, almost like harassing the females. I know that's not like a scientific term for it, but they're kind of just like, looking for their females, trying to settle down into their territories. And yeah, it's a lot of like sitting and watching, and you might have to like later in the season you're gonna be like, okay, like now I have to find the nest, so you're gonna kind of sit back and look for where if you can get eyes on a female with your binoculars, where she might be, looking around like, oh my gosh, gotta get back to my nest, gotta sit on my eggs, gotta feed my young. And then once you're starting to see more of those behavioral instincts from females looking and monitoring, like almost just like going back and sit and hear the grasses and the nest and extension. So that's lots of times we couldn't find a nice sorry. They're very good at his job. No, it's with the fourth. Yeah. So yeah, finding those nests were very true. Yeah, and once we're like identifying the nest and once they're starting to lay eggs, tracking like how long until the eggs are going to take to hatch, and like how long need to wait until they're able to take the babies out temporarily to get like vital information such as their weight, or how old they are, give them their identity through the colored bands that you see exhibited on the legs. Taking blood samples, which is really interesting to me personally. I was not expecting to be sitting out in the field at like 9 in the morning and just like casually taking blood samples from birds. And as we like to know, putting the little nestlings in their little burrito to make sure that they're not flopping around and we get their accurate weight. So our summer data, we technically had 5 nests with 1 exception. We had a bunch of different pairs of birds that were notated by the 3 colored bands on their legs, along with a steel band that gives them their number identity. And throughout the summer, we were able to notate like how many eggs they had, if they were decrayed, abandoned their nest, how many nestlings they had once the eggs hatched, confirming fledglings later in August, and getting like specific GPS coordinates to track exactly where their nests are going to be, in order to compare it to last year data from, I think Nikki presented last year about her research. So I was continuing her ongoing her research, and it was a lot. Like we did have a lot of success this year. We did have 1 nest that completely abandoned her nest. However, later in the season, when we were started to slow down and really start paying attention to see when the birds were going to start fledging and tracking that, which we did pursue all of the nestlings did fledge. We did have another renest, which is really weird for bobolinks. They don't typically do that. Weren't able to find the nest that the babies came from, who were the male, but YOGR female did have 2 confirmed fledglings after her previous nest was abandoned of 6 eggs. In the map on the right, we have the different territories of our birds. So we did have 5 successful territories. In addition to, you can't really see it for tourists in my part, there's a little purple dashed circle in the left-hand, or kind of middle left side of the map, where we did find a lot of other females that were abandoned, presumably coming from possibly nests from other areas that were just kind of scoping out this area, seeing if maybe they could get in on some baby action or something. And yeah, there's a lot of walking around and watching. Definitely teaches people a lot of patience. So here, the difference in experience is established this year. Was one of them seeing that and that's so it's a kind of explain what we do. There's a bill on this side. But last year we had that's possible. And this year the male that was responsible for this has not nested at the end of the field. This year he actually established different relationships with different things. So he actually that's one of those babies that was invented. And so obviously the territories we're looking at the different events there. Yeah. And some ways or one sample top of the field that's your and so we kind of at the top and stayed nesting here last year nested in that so that is LGBT and so yeah that was their nest had the different egg and they were so they kind of maintained where they were last year in this area which is different from the company that split is one of those things is really big that's very so in addition to having bobolinks, we've also found some spheres, which they are very similar to bobolinks. The grass and birds, discussions, however, the spheres are not being that's the appropriate option to run it but I'm sorry to change about wetland. So that indicated that they have successfully successful production. And the fact that we find that this hayfield considerable problems and transfers suggests to us that we need to survey this on hayfield because of their bobolinks and transfers. Being able to put it with their this year, any case of this and going back to all the bird loss receipts, some of these very different. So maintain the population that if I have say at home services, we're not so much yeah, and we only have 5 years to extend all the biggest windows we have to cross really great to see about this. So it's coming down to not so this is very important. Please So for next steps, we really encourage people, to really have more students, coming back each year to continue Nikki's original idea of surveying site fidelity, productivity of bobolinks, and even potentially monitoring the populations of the savanna sparrows, because as specialists who are a bit more picky about their the types of foliage and other nesting materials that's around, having like Julie said, having the introduction of a new species in these hayfields that are recovering from the previous agricultural fields proves to us that succession is really moving forward and giving that that more natural what the habitat used to be. And we want to encourage minimalizing the differences about the disturbances to the grasslands. We did have an issue which might have led to some of our abandonment of prey nests of a dog was on a leash in the fields, unfortunately. And we did try to, inform the lady in charge of the puppy that, hey, we're doing bird research here in the field, and your dog running through our fields is kind of puts a brick wall up for us. But we don't really know exactly how much that might have influenced any of our bobolinks that we are monitoring, but we did still have a lot of fledglings and successful nests. So just encouraging the decreased disturbances would be something to keep. We just want to thank RCA for having us and allowing us the undergraduate students to conduct research and really get our hands on and work with, Baldassarre to the research of us biology students to really have the ability to exercise our interests. Thank you.
Showed your maps, they weren't all circles, they were different shapes.
Yep.
How do you determine those shapes?
They are rough, just estimates based off of observations that we made monitoring the behavioral behavior given off by the birds, just monitoring where they were throughout the year and making notes of where we might find them. We were running a research from about May 18th to about mid-August. I don't have the exact date in mind. So it was just a lot of just sitting around and being well, Julie, how about you take a watch this half of the field, I'll watch down here. And then also having Baldassarre's help too.
It's like where they're feeding and things like that.
It's kind of just where we're watching them land and fly around. So it kind of isn't, it's just mostly an estimate of where we might find the birds, but it's just more of where we're observing them flying and thinking, especially just early on in the season when they're just picking out who they're gonna have babies with, and yeah.
Then are the fields mowed? Is the hay used for? Is it hay? Is it being used for anything?
So it is not necessarily used for anything. We do also run into the issue of cross country runs in early fall through those fields, but due to NCAA standards, unfortunately, there are a lot of large strips fragmenting through the fields. So we do run into that disturbance, but that is not a fight I can fight, or Julie can fight. So even if they're not running that during the summer, it does kind of fragment the fields, leading to less opportunity for what space is available for the birds. But we did see an increase in bird numbers, so they are pretty happy with the field as is. But it is a little frustrating from the birdie side of things.
Well, we made great progress in management of the fields. The fields used to be at least 2 acres. So the birds would nest in the field, and then the hay would get harvested exactly. And so administration stopped these hay farms. Just they still do get mowed late enough in the season to keep them open grasslands, but now they go to a certain. So all in all, despite the fact that there are disturbances like the dogs and the fact that the cross country trails, it's much better shape.
I think in areas where there are signs during the breeding season, and you don't have your dogs there, or you certainly don't have.
Yeah, and that it is a blanket policy across all these fields that dogs are supposed to be on a leash. We could do better about having signage up.
Well, no, they're taking the things.
Yeah, we should try to do that because it definitely is a source of disturbance.
I was very interested in where you were finding the bobolinks, because I ain't never seen them.
Oh, they're so
very
fun. They are very sneaky. They're a lot of fun. I would encourage you if you have the opportunity to, like around mid-May, early June, trying to get out there. Even if you don't have a pair of binoculars, it's pretty easy to spot the males because they typically are singing their song.
Not that I know where they are. I'll be.
Yeah, and they have like typically like a nice white cat.
I've seen them, but not
So,
so years.
Yeah, they're out there. It takes a couple tries to really get eyes on them, but they're, they hold a special place in my heart. They're really sweet.
How did you go about safely finding these nests?
So it is a lot of tedious work. Those were definitely our longer days. I would like to use the term or the phrase frolicking in a field where you're technically sitting back and really watching for those females, watching for feeding behaviors of her potentially bringing worms or insects to what might be a nest, or she looks typically very suspicious, like she's just itching over. She's just kind of like looking around, making sure nothing's following her. And once you're starting to really watch her and like see her dive in, if she's not coming up for a few minutes, it's like, okay, like that general area is kind of, an area of interest. And typically when you're going into the field, you're gonna want to be very careful because they do nest very close to the floor. So stepping in areas that you're ensuring isn't a nest, which I know it sounds very big, but just watching where you're stepping. And then we bird people like to use the word flush off the nest. So just watching for where that area of interest is, and as you're getting closer to it, watching to see if you're getting really close and she's like scurrying off the nest and you're like within a couple of inches of her, you're gonna want to look and you're just gonna be like going through the grass and looking. They do like to really hide in there, but I was actually the one to find that one nest, which, was definitely an interesting find. And it's just you can kind of tell there's a lot of like leaf over it, so just ensuring that you're not searching for hiding because no one wants that. And just like slowly moving through the grass. It's a lot of just trial and error of trying to look for that flush, and if you're not seeing it, taking a step back and watching for 10, 15, 20 minutes and watching for the behavior of her diving and then going back in. Because the males aren't super involved until the babies are a bit older. So it's mostly just really practicing how to identify those females.
Around here, is there many predators after the birds? It's not the bird, the adult birds are smart enough, they'll fly away, but it's the babies that the predators will attack. Am I get that right?
Yeah.
Is it a big problem here? I mean, between raccoons, feral cats, you can say all sorts of animals that go after the birds.
It's kind of a loaded question just because, you have your natural factors of possible snakes, like wanting to bite, or raccoons, or even corvids. We did see our one nest was the prey of 2 little babies that were, just hatched, maybe a day or 2 old. So I feel like, to my best of my ability, it would be any of your natural predators who are just for a quick, easy meal. That might consist of, snakes, raccoons, corvids, anything that might be natural predator.
Yeah, between the hay farmers and the predators, the birds got it tough out there.
They definitely do have a tough, which again, goes back to why this is so important in monitoring these, especially since we can use MAC protocol. So we can send our data out to other people and see what they might be finding, or if we have any questions like, what the heck is this random egg? And just trying to see what other people are up to really analyze, worldwide where all bird populations are at, whether it's big grasslands or big neighbors, forests, and the process. It definitely can be a factor, which is why when we're doing nest checks, we really did do nest checks we really didn't stay around the nest for very long. We just quick glance and walking out of there. And we also ensure that we had a GPS location that way we can take different ways into the nest to ensure that we're not making like one trail to really minimize that. I think we did nest checks once every few days to really, and like once the babies were hatched, we really slowed down because we were like, you don't want to come on and see that it's not to make their young and be a factor to that.
Is your data used for like further investment conservation?
So the data right now is just like for us specific. For like, sorry, whoever he chooses to continue the research next year. Ultimately, the longevity of research, as we see with having the longer, like the more years under our belts and seeing how the patterns may shift and change is really what we're looking for right now.
Presentation is by Dr. Weaver from Atmospheric and Geological Sciences Department. So we have J.R. Slosson with the study of the actual creek of Rice Creek.
As Andrew mentioned, I'm in Atmospheric and Geological Sciences, so I am more of a water person in this perspective, with a presentation that says biology adjacent. So my interest here is really in the wetlands out here. Before you go into the specific research question, I'll just give you some background. So historically in the US, we've tended to view wetland types of terrain as sort of like nuisance terrain, most like places that we don't value, want to fill in for it to a better type of land use. I think attitudes towards that are changing, but those tensions are still there. But we're starting to realize more and more is that wetlands provide all kinds of valuable ecosystem services for us. That includes attenuation of potentially harmful pollutants. So I call nitrogen, phosphorus in here, pollutants. They're limiting nutrients for plants, so they're good until we have too much of those things. So we'll use them in fertilizers, we use them in lawns, we use them in golf courses. If we use a lot of those things, they make their ways into our rivers and streams. That can be a problem because they get to things like harmful algal blooms, which are problematic all across the country. Wetlands are useful because they attenuate concentrations. So plants assimilate forms of nitrogen. Bacterial processes also. So folks have started to catch on to the fact that wetlands do this and have tried to mimic those processes to what I loosely call constructed wetlands. In many cases, these are like engineering types of structures that folks will install in their agricultural lands and things like that to try to replicate what natural wetlands. Okay. Rice Creek is sort of in an interesting area. So the hydrology that we see today isn't entirely natural. It's created by impounding the creek just to the north of us. So we created Rice Pond, which helps create the natural or the nice wetland environment that we have today. Right? So this is kind of an interesting scientific experiment, just this impoundment that is used to create adjacent wetlands, replicate many of the features as what I would call an India. All right, before we talk about what's going on here, we should talk about potential pollutants or things that impact water quality entering fields like here. Okay. This picture here shows the Rice Creek watershed. So the black outline shows the entire watershed, something like 30 square kilometers. Fulton is down towards the bottom of the map, just off the map. So it's big. Things that can affect the water quality. So it'd be geology. So dissolution of minerals and these types of rocks that are common in the watershed can make their way into the water. So you've got sandstones, you've got siltstones, you've got shales. So we might see signatures of that in our water. We also have different land types. So we see a lot of deciduous forest. That's not surprising. You've got hay, pasture, cultivated crops. These come from the National Land Cover database. So I would categorize those as agriculture, broadly speaking. Those places might be a source of excess nutrients. We do also have natural wetlands, all Gulf watershed. We've got a mix of both of these. I put up here too low intensity developments. These might be things like roads. We'll see how that might play in with water. Okay. All of those things enter Rice Creek. They flow down past Fallbrook and then into the pond here. So this is aerial imagery that's courtesy of Liam Todd and my department. So this is late summer, late August, early September. So you've got the pond, so that could affect water chemistry. We've also got the marsh type of environments at the margins. You also have more marsh as you get above the bridge. I can't talk, but I presume most are green colors. We are going south, so the bridge where all the construction is will show up as. Yeah, we're going much.
There are marks in the water and on bus scratch or something.
I don't know what those are. So I mean, it could be boats even. So someone may have been out there, got the boats back there. I've never taken one of the boats out myself. Yeah, I mean, just above the bridge, the beaver dam was reestablished for this subdivision. So we'll zoom out. The bridge is basically just around the corner. You can see the Fallbrook property as we get a little bit higher. Now you've got more marsh to this part too, and the boardwalk.
When did you say this video?
It's either early September or late August. I can't remember.
Is it?
This year. All right, so some goals of the project. So first of all, before we think about what role the pond or the adjacent marsh might play in changing stream chemistry, we should just take a look at what the stream chemistry looks like. Okay. So the way I approach this is by looking at major ions in water. So these are going to be products of rock weathering primarily. They can come from biologic processes. They might also come from human related. Okay. Ions they're just charged molecules. Okay, so these are things like calcium, magnesium, chloride, nitrate. So those all might be. Okay. We need to decide where we're going to collect our samples. So we collected 3 different points. So we collect. I'll show you on the map in just a second. At Fallbrook, at the bridge, and then at the outlet. We also sample 2 groundwater wells roughly once a month. Groundwater velocity is very slow, so I don't expect the groundwater chemistry to change very much. But we want to get a sense of what's going on. We use equipment in Shaylynn Science Center to determine the chemistry. And the other thing is important too. So the concentrations of any of these ions is also impacted by how much water there is. So I also have two water level loggers that monitor how high the water is. This is me and my team. So this is a lot of work to collect all of the samples. I did a lot of this over the summer. Students have mostly taken over. Okay, these are the different locations. So we've got a surface water site at Fallbrook, which will capture the geochemistry of water entering the site. You've got another site as you go through the marsh next to the bridge. And then you've got another field site below them. So 3 different places. You see all the chemistry. And then we have our groundwater wells too. So you've got one closer here, which I've just lived with the Rice Creek well. Another one over. Okay, let's start with the water height and what's going on with the precipitation. So it's a very dry summer. Okay, so in July, and especially going to August, it basically stopped raining. The rain that we had was very, very small and quantity. And we can see that replicated in how high the water is at Fallbrook and Pond Outlet, basically just dropping also. Then it starts getting rainy. Okay. I won't show all of the chemistry data I have because there's quite a bit. I will show some highlights because this project is really just starting to get going. We anticipate going from. This is fluoride. Okay. I don't start collecting from all 3 sites until June. So there are a few sites I sampled before that. We get all 3 starting in June. Fluoride could come from geology. I don't think this is the case here. But chloride broadly represents what happens with some of the ions too, which is that their concentration gets higher as their summer goes on because they're just less. All right, so then the question is, where's the chloride coming from? One thing we can look at to take a guess at where the chloride is coming from is the groundwater. So chloride concentrations at the Fallbrook. Chloride concentrations at the Fallbrook well, they're pretty low. They're less than 5 milligrams. That means for every liter of water, you have less than 5 milligrams of chloride. If we look at the well closer to Rice Creek, concentrations are much higher. It's like 80 or so. The concentrations in the streams, they were lower than that. Okay, so let's go ahead. This would be consistent anyway with folks using road salt out the highway. That road salt entering the groundwater system and basically slowly migrating down gradient towards the well dip. Road salt stays in the groundwater system for a long time, so I would expect down gradient wells to have higher concentrations and to continue to have higher concentrations. That will slowly seep out in the creek, elevating. One way to know for sure is see what happens in winter. So I intend to collect an example in winter, but we'll see what happens. I don't necessarily mean to alarm people, so some comparisons to other places are useful. EPA's chronic ambient water quality limit is 230 milligrams per liter. So EPA has basically said that if you're exposed to these levels and you're an aquatic critter for 4 days, that's not. We're not at those levels. And just as a comparison to a much more urbanized site, these are data from Syracuse. This is from Meadowbrook Creek in Syracuse. It's representing lots of different creeks in the Syracuse area. Their concentrations, even in the summertime, hover around 230 milligrams per liter. Fall summer. Then there's thousands of them. So that's not our situation. I would say our situation is common for this part of New York. Concentrations like those. I've seen concentrations like this even in like the buildings and periods in the Adirondacks here. So any place where we use road salt, we expect. Nitrate, so you've heard about nitrate. The nitrate result was pretty interesting because this is where our sites actually start to do different things. So we've got nitrate concentrations coming in from Fallbrook, which are usually higher than the other sites. As we get into sort of July-ish, the nitrate concentrations at the pond outlet slowly drop beyond the detection limit of nitrogen. Nitrate concentrations at the inflow, the bridge up there, remain elevated for a while, but then they start dropping. So that suggests that something is happening to make the nitrate go away. Two likely explanations that we see with like wetland type of environments. So either assimilation into plants, the algae. If that's the case, I expect to see some of that nitrate come back. Another possibility is denitrification via microbial processes, in which case we won't see that. We've been lost to the atmosphere because nitrogen gas. So we won't know the full story. Let's see. All right, this is the result that was, I would say, unexpected. Sulfate concentration. So sulfate, like chloride, all the sites basically look the same for most of the spring and into the summer. Once we start going into late summer, when it's really warm and we've had drought-like conditions, sulfate concentration started dropping at the pond outlet. A possibility is that you have sulfate reduction by microbes in the sediments in the pond. But then you get this really big spike, which is what I didn't expect to see. This might be because of the drought conditions. So it could be that you've dried off soils, got a lower water table, you're seeing oxidation of sulfide minerals somewhere in the watershed. Then it finally rains and brings that sulfate into the juice. Other folks have noticed a similar trend after drought in the Northeast. So that's probably my best guess. But it's all still early days of the project. So far, Rice Pond probably is a meaningful sink of seasonal nitrate. Groundwater surface interactions show signs of road salt contamination, but I would say the magnitude of that road salt contamination is similar to what we've seen in the and then we see the spike in sulfide, which could be. This project will continue through the winter, so we'll see what comes out of that.
Your nitrate expectations, I guess I'm a little confused on why you expect it to come back in fall. I'm assuming that the input is largely agricultural and lawn runoff, right? And people stop putting that on their lawns in the winter. And if it's taken up by the algae, I mean, they're gonna die and then it's ammonia or something, so.
Yeah, usually we see nitrate concentrations come back up in the winter time as plants die. I mean, that's my expectation is
Even if like the input kind of slows down a little.
yeah. And the inputs I don't expect to see a lot from agriculture.
Then for the sulfur, no possible like sulfur oxide adding bacterial gases.
I mean,
I guess I'm looking at the data differently than you would.
right. I mean, I suppose I can't rule that out either. But I think about it makes sense connection. So.
It's probably geology.
That's my guess, and it's still too early to really.
Question today. Would you say that this created wetlands is performing a lot of the services that are not performed, like?
Yeah, it certainly seems to be in terms of at least the nitrate see-throughs. So it seems to do that. Yeah, I mean, one thing too, we don't have the amount of nitrate coming into the system is not as high as if we were connected, you know, to a large scale agriculture. We have this input. So it's hard to estimate the total magnitude. But we do certainly see replication. I would say the concentrations that I see here are lower concentrations that I've seen in the other systems. Well, I don't know.
So you say, I'm sorry, just another thing about it. Say that water becomes just takes its time to go through the wetland system. You say it takes its time, right? Time scale that you're talking about.
Yeah, so
I
it
know
depends.
it's very
So
quiet.
I think there are a couple different ways of thinking about this. So within solutes of any kind, just moving through the surface water system out here, it seems to be pretty quick because the concentrations are close together all summer long. We don't see significant deviations, which would tell me that there's a longer than this time. Groundwater is much slower. So to get from, for example, you know, the road to the stream, depends on your subsurface properties, but it could take months to decades.
Hey, this is really interesting because you're just using a subset of a creek here. But in the future, is it been done or do you intend to do it for the river system? A much bigger. I wonder what you're gonna find there. Okay, what would you expect to find? What I'm gathering is just a novice. This is a happy place. Overall.
Yeah, I mean,
And we like see that for the river.
something like the Oswego has higher concentrations of everything, basically. So, yeah, that's a very different system. It drains Syracuse too. So, it looks the chemistry looks very different than some that's most.
We live on, place over. West of your program.
Yeah.
And that absorbs water. Then it leaches now. So we have first some problems.
Right.
Is that?
I mean, there's certainly surface groundwater interactions. The way, I mean, one way to know for sure would be to watch some stream temperature, see if it stays in the time. That would be one way to trace where your sources of water come from. But I suspect that this is a discharge point of probably processes that are from ground. I mean, usually if we don't know what the subsurface hydrogeology actually looks like, we assume it mimics the surface topography, which slopes down to. Yeah, I mean, one thing I didn't put in here is the level in the groundwater wells. I mean, they've been dropping all summer.
And as a little pitch again, if you're not a member, please pick up a brochure and give it a consideration. Thank you, everyone. And we will be doing a letter fundraising campaign for small grant. And I need a group photo of all the presenters. If anybody knows.